My name is John Cousins. I'll just fill you in the backstory a little bit. The company's Biomoda. It's a biotech company. We're developing cancer diagnostics. Our first test is for lung cancer, and it's based on a molecular marker that binds with cancer cells and fluoresces red under ultraviolet light. We're using sputum as the sample, which is the deep lung fluid count. We also work on basically on any cell sample, any body fluid sample. We're looking at getting body fluids that are easy to get out of the body so we can make non-invasive tests that then can be used on a large scale for screening cancers. Lung cancer is the largest cancer killer, and it has the most dismal survivability rates. Only 15% of the people survive 5 years. 60% die in the first year. So there's a real need for an accurate, inexpensive test. There's no current screening protocol available for lung cancer. The standard of care now is CT scans, which are relatively expensive and rare assets. People don't go on a screening program to them. Basically, that's used when you present with symptoms, and the symptoms are pain in the chest, shortness of breath, things like that. That basically means that you're already at the end of the lung cancer development process, stage 3, stage 4, and at this time it's pretty much a death sentence. And also it can't really resolve tumors less than about a centimeter, and so you've already developed a tumor by the time CT scans can resolve where it is. PAP cytology has been very prevalent in the cervical market, things like that. It's also a way to identify other cell samples, and it's used to a degree with sputum, the lung fluid, but it really doesn't have very good, strong different therapy routes that become available if you can catch lung cancer early. One of the most successful is resection. It's actually in a small area. Resecting a lung of the lobe has been really, really significant in long-term survivability. But you have to identify it before it spreads. And there's a number of other things coming just on the market now in genomics and proteomics, pathways of proteins and the gene development of those proteins, pathway inhibitors, so you can limit the development, the oncogenesis, the development of the cancer by actually limiting the pathways that develop the fertile ground for the cancer. One of them that's really prevalent right now is a pathway inhibitor that inhibits the ability of a small tumor to develop more blood vessels to it so that it can actually grow. So as those develop, the early diagnosis becomes more successful and efficacious as well. So we're really in a nice time here to be sort of complementary with the developments in various therapy routes. This is a depiction of the molecule. It's like the heme and hemoglobin. It binds to cancer cells. It's a surface binding mechanism. It was originally developed at Los Alamos Labs. Biomoda initially did a licensing agreement, and actually before that a CRADA, a cooperative research and development agreement with the national labs to do the development and the technology transfer. Since then, the company's developed a owned patent portfolio around this molecule and the techniques for cancer things. So we started out with a licensing agreement with Los Alamos and migrated into a owned patent portfolio that the company's developed. This is our bottle of our compound. We trademarked the name CyPath for cytopathology, and that's what we're calling our lung cancer diagnostic. As you can see here, a couple pictures. There's a cancer cell under white light, difficult to identify. Under black light, labeled with this, it glows red. It makes it really easy to automate this process. Right now, the art and the nuance of reading for a cytopathologist to read cancer cells with white light, it's a lot of art. It's not a simple process, and it takes a lot of human overhead, experienced overhead to read those. With our system, the ability to say, is it glowing red or not, that kind of a binary assessment makes it really easy to automate this process. So you can screen large populations if you can automate the test. So
which is the cancer cell? There's two masses over there.
On top?
On top, yes.
It's this cell here.
Okay. Both parts?
Yep.
Okay.
Is that the same slide?
Yes. Yep. Same slide.
Another thing I'd like to add is that what we're running into, and Connie can talk to this, is that there's varying degrees of how red it gets, how illuminated it is. And what we're finding is it may be that we can, and we're not positive of this yet, but that we can find it in very, very early stage, and this would include pre-tumorous. So there'd be a pre-stage 1 diagnosis of potential for cancer, which would make the lung cancer survivability, one would assume that would really make that go up. And this test is really cheap. It'll be really cheap for everybody to have, and less complicated than a normal blood test and a physical. So that's the possibility. We're trying to dial in where our thing fluoresces red, at what point we have a cell that's potentially or is cancerous, and you put in these really primal early therapies at that point, and then you'd have a whole new kind of curve of who survives lung cancer and how they survive.
Capture the images. It's a microscope. You put the slide on the microscope with an ultraviolet light source. We use a new sophisticated image capture device that can actually, it's a narrow bandwidth that just looks at the bandwidth of the signal, the fluorescent signal from the TCPP. And then we actually capture those images, and we can do them in multi-layers and things, and then analyze and look for cancer, basically, and it is the level of fluorescence, as Tim said, that is indicative. Here's another picture where you can see how difficult it would be to spot a cell. Here's one cell glowing red. There's a normal. You're really looking for needles in a haystack in a lot of instances, and to be able to. And this is a field on the microscope at 200 times or so. Just one microscope field. This image is probably about 1/40th of that entire field. So as you go and search over the whole thing, it can be very difficult to find. This shows it up images very easily. And here's a picture that shows the surface phenomenon. It's the internal proteins fluoresce green, and on the surface it's fluorescing red. It's labeled with our color. We're in our phase 2 study, our pilot study, and we are collecting from veterans that have smoked 20 years, 20 pack a day, 20 years. That's our high-risk group that we're screening. We're doing that in New Mexico. It's actually been funded by the New Mexico State Legislature. And we are collecting for every patient a CT scan, which is the radiology standard for identifying lung cancer, and the pap cytology, and then our test. And comparing the three, we did the study design, ran it by the FDA. They came back with their revisions, and so this is our study design that they would say was acceptable. We're wrapping this study up in the next probably 2 months, and right now the results are very strong. Then we'll ramp up to our phase 3 study, which will take us basically the bulk of next year, submit to the FDA and get FDA approval, and then we can actually sell this on a large-scale basis. So that's our regulatory development path. We're putting a longitudinal component in our study. The ones that we've identified that look like they have early-stage lung cancer, we're now doing follow-ups on them every 3 and 6 months to see how that cancer develops.